Hopefully you guys can hear me. I'm getting over a cold, but my name is TJ Cradick. And as mentioned, we're going to talk about using nucleases, primarily zinc finger nucleases and tau nucleases, or TALENs, to target single gene diseases. But if you don't work on those, you can put in your head whatever you work on, because your favorite gene can also be targeted. I'll give a brief overview. First, why we use nucleases, and then talk about nuclease design. And in the case of TALENs, the DNA binding domains, there's only a few choices. So it's more a question of picking the best or the optimal target sites. I'll give you a little background about our research and how we do testing and validation. I'll start off by talking about different kinds of genes that are disease genes. In the case of something like sickle cell, there's a primarily mutation, or cystic fibrosis, one spot that a lot of the patients have a mutation. And we can go in and edit that one area and get a lot of the patients. But other genes, there's all kinds of mutations. There's patients that show up that have missense mutations, truncation mutations. And in those type of instances, if we're trying to repair the gene, we can't just target one spot and get a lot of the patients. So therefore, we might want to replace the gene and throw in a cDNA. But in either case, we're editing the gene to correct the mutations that occur in patients. And that's how we move forward. But to knock it in, if we have a DNA break, we get about 1 million-fold enrichment. So, instead of screening millions of cells, you can get up to where you can look at a very small number. And to do that, we need a very specific nuclease, because you obviously don't want to cut other spots in the genome. And so to do that, we have a non-specific catalyst that cuts from a bacterial endonuclease. But we target it with a very specific DNA binding proteins. Zinc finger proteins, which I've drawn here, make zinc finger nucleases, and tau effector proteins, which are the DNA binding domains to make the TALENs. And so if we create these, we can target spots, we can engineer them to go to your favorite gene or our disease target. But key to the specificity is it takes 2 to bind to make the dimerization domain of the FOC. So instead of targeting just one spot, they occur much less often in off-target applications. So it's just a lot more multiplicatively more sensitive or more direct, because it's targeting all of these bases. One thing I want to emphasize, though, is in this case of the zinc finger here, it might be targeting a G, but it might also, to some smaller degree, also bind if there's a C there. So in each case, for both the tau nucleases and the zinc fingers, they're not exquisite in their specificity. And that's one of the reasons, and one of the projects we have undergoing is looking at off-targets, and just to keep a sense of how much they could go to other spots. And once we cleave the gene, depicted here, there's 2 main classes of cellular DNA repair that the cell can undertake. The down and dirty is if the pieces kind of come together, and they generally have a lot of insertions or deletions. And that's referred to as non-homologous end joining. And so if we're using that, it tends to be when we want to knock out a gene. If we want to see the effects of removing a gene, or we want to knock out a gene, they're using this type of approach, principally with the CCR5, where they're taking the T cells out in clinical trials and removing the HIV receptor. They just want to knock it out. They don't necessarily have to edit it. But if you put in a donor DNA, and this can range from oligos to much larger, that has a mutation change from your site, once you cleave, homologous recombination can edit the gene to be as you wish. And you can knock in a full cDNA, as I mentioned, or you can put in single base substitutions or deletions. And so, depending on whether or not there's a donor and what type of cellular repair determines how it's repaired. We've got a number of different targets. As I mentioned, there's gene therapy, or instances of reverse genetics. You get a patient, you wonder how important that mutation is. Or if you want to modify cell lines to express a lot of a protein, or to be a model cell line, or animals. And you can take C. elegans, you can take zebrafish, mice, rats. We've done a whole number of these. And we're looking to do others, and you can make the same kind of genomic modifications in these animals. And the cells that you target is also can be varied. Cell lines are one of the easiest things if you want to modify CHO cells, 293s, just to test your hypothesis about your gene. For gene therapy applications, we want to target the affected cells. If we're trying to correct the sickle cell mutation, and obviously we want to target the blood cells. But we can also target progenitor cells that give rise to those cells, whether pluripotent, embryonic, or other stem cells. So this is where we can also take a look at, well, how many do we need to repopulate the cells that we want? And so there's a lot of things that are going on in exactly how to deliver these types of nucleases. And one of the things that Wilbur Lamb's lab focuses on is trying to deliver. But in our section, we primarily work on designing these nucleases and monitoring their efficacy and off-targets. And in this regard, we want to get the highest activity, but the best specificity. And so we changed the design criteria, and as I mentioned, possibly the target sites. And so to measure these, we have a number of different assays. The simplest is an in vitro cleavage assay. And this is basically as easy as a restriction digest. And I try and tell ages ago when we first developed this that you can come in, have your DNA linearized the night before, add some express protein, and have data to present at a lunch meeting. And I met these guys that I taught the process in Germany, and I didn't find the professor at first, but his students all knew who I was, and it was because this technique was really fast and easy for zinc finger nucleases. A little harder though, as you go in, you have to do tissue culture. Obviously it takes a little longer than the morning, but you use the same kind of target plasmids and you see if you can cleave and possibly repair the target plasmids. Then once we've narrowed it down, then we tend to go and look at the endogenous targets. And as I show in the last part of the slide, we can look at these both for activity and for specificity. So I'll start telling you about one of the assays, just to give you an idea, where we look at the target plasmids. And we're looking at something similar to homologous recombination, but it's called single-strand annealing. And so if we have a target site cloned in the middle of the GFP gene with a little bit repeated, we have a stop codon, so it doesn't express in the uncut state. But once you have the nucleases cleave, it can be repaired by non-homologous end joining, and then a PCR product will be about that size. But if it combines in something similar to homologous recombination, namely single-strand annealing, the two repeats get left off as do the stop codons, and you have a much smaller PCR product, but you can also observe the cells are green. And so we did this in a large scale, and so we moved into the high throughput means of doing it by PCR. And we transfected 293 cells with this target and with either one or pairs of nucleases. Then we harvested the plasmid DNA, did the PCR as I mentioned, and then separated these on a gel. And that was to derive rules of TALEN design. And so far in the literature there was only a couple guidelines about how to make these. And we thought, well, maybe we can find out what correlates with good activity. So if you took your favorite gene, you could figure out what spots to start with, what had the best potential to make really good nucleases. So we screened 100 and something different TALENs using this assay and solved to find out what had the best activity, what features, what nucleotides, what length, we went through a whole range of different features. And we made more, developed these rules, and the graduate student in Dr. Baza, Nani Lin, did a lot of this work. And we're talking about hundreds of these molecules that in the literature doesn't really have that many at this point, so a big chunk of them. And we've compared the scoring to their activity in this SSA assay. And we're very happy that it's moving forward. We're about to finish this off and submit it, I think, in a day or two. So I hope to be able to share more of this story when we go forward. But we were able to get some TALENs with near record levels of activity by picking the sites using this method. And the way it works is you input your gene, and you can have it graphically show the activity in relation to the distance to the mutation. And it gives you a ranked list of the target sites. So you've got the sites that and you can just use those to make your TAL nucleases. There's a program we throw into our liquid handling robot and it starts to assemble them. And here I've annotated a couple spots. And so the second highest spot is right next to your mutation. And you want to get as close to the mutation as possible because that increases your rate of homologous recombination. But normally in cells, it doesn't work exactly like it does in the plasmid assay. So it's kind of usually good to look at a few different sites. So we might take the one in red and the one in green and a few others and synthesize them and see how well they work. So this is going to be a website that will allow other people to do the same type of thing so that they can also design high-quality talent nucleases. And then you take them and test them in plasmids and then go on to endogenous genes. And so in the write-up for the paper, we compared to one publication that also did about 100 different nucleases, except they used the DNA binding domains that are a little less specific. And we decided we'd rather start with the ones that are more specific and see if we can increase the activity going through all this work. And so we were happy to see that our average activity, around 41%, was higher than theirs. And then, I'm not sure what we were thinking at this point, but we wanted to go after the really hard ones, the 12 that they got no activity on. So we targeted 4 out of 5 of those successfully. So we were fairly excited that we were able to use the more specific type of DNA binding domain and then increase the activity. And then the hope is that we have both the highest activity and retain the specificity going forward this way. And as I said, we're hoping to submit that that might have gone today. The ongoing work is that we're going to expand the ones in our test set, trying to improve the correlation. And then we're going to test the ones that look good in this activity towards the endogenous genes, and we'll use the program I'm going to talk about next, and just see how well these work in the different applications with our different collaborators who are happy to have a way to modify their genes. The second major project was done with one of the graduate students, Eli Fine, and this is another website that we're trying to provide to the community, which ranks and tests possible nuclease off-target sites. So once you treat your cells with either the zinc fingers or the tau nucleases, you want to figure out what are the usual suspects? What's the best place to look for possible off-target activity so that you can look there in great numbers instead of sequencing the whole genome, which you can only do a few times. So once again, we wanted to make a good user interface that proved an easy way to do this. And what we've compared it to is other methods where they characterize the binding of the nucleases and then try to use that to go forward. And in this method, if you can transfect cells to do PCR and send off for sequencing, which has gone way down in price, then any lab doesn't have to learn all the expertise of the assays that we were developing that were never near 100%. So this is the website. You enter the target sites of your particular nuclease and some of the specifics that determine which genome you're searching in and how you want to run. This is one of the activity assays that was formerly often used, but we're going to move towards the sequencing idea. And then it kicks out, and this is from an old website of mine, that gives you a list of the similar sites in the human genome, or whatever genome you're looking at. And we often would attack the top 16 or the top 24 sites. And most of the time, even with ones that don't work that well, you only have a few off-target sites. So once we've identified the genomic sites, the program also kicks out the PCR primers. So you don't even have to design the PCR primers. And the good thing is that we have the liquid handling robot do the PCR, and it works at over 93%. So really straightforward, high throughput way to PCR all these sites and pull them and send them for sequencing. And so if you look at the target site and all the off-targets, you can compare the relative activity at the target site and off-target. Similarly, you can test different designs of the nucleases, different treatments. If you want to modify some DNA repair enzymes and see how that affects it, whatever you're trying to study, you can then go back and look at how effectively you targeted the target site versus off-target sites. And this works both in tissue culture and in the model organisms that we talked about. And so we've done it now, we've looked at different types of zinc fingers, and we're able to find out the kind that's the most specific. Similarly, we compared different sets of tau nucleases to pick the ones that were the most specific. And we just completed a collaboration with a group that makes these knockout animals, and we wanted to confirm that they didn't have off-target sites, which would require a lot more breeding. So the ongoing work is we're going to design more of these and continue testing. And one of the things that I wanted to mention is that you've got your favorite genes, let us know, because we can add them to our target genes. We're particularly interested in developing nice cellular assays, or maybe if we can modify a gene that affects how they squeeze through, we can go forward. And also ways to deliver these. It's another ongoing issue. I'd like to thank the lab and a few other people that I didn't mention, and Dr. Bao. And with that, I'll take questions, please.